Hello and welcome everybody to my channel Omini Bio Entrance, and today we will be discussing GATE Life Science question paper conducted in the year 2023. This is for the biochemistry section. If you are new to my channel, please do like, share and subscribe. Coming to the first question. Determine the correctness or otherwise of the following assertion and the reason. Assertion: on a per carbon basis, palmitic acid yield more ATP than glucose. That is correct because complete oxidation of palmitic acid will give you 106 ATP molecules, whereas for glucose it is 36 to 38 ATP molecules. Now why does this happens like this as per given in the assertion? The reason is carbons in palmitic acid are more reduced than in glucose. That is also correct, and it explains the assertion. This means that they have more electrons and can generate more ATP. So answer is both A and R are true, and R is the correct reason for A. So answer is option A. Then next one. When cell components are fractionated by sedimentation, the correct order from lower to higher gravitational force in which the components get separated. So during sedimentation, the order of separation will be based on size and density of the cell. So the one with which is largest and most densest out of the given options is your nuclei. So first will be nuclei, then the last one will be, which is the smallest and the least dense, it will be your ribosomes. So if you see option 1, you can see that nuclei is the first one and ribosome is the last. So most probably that must be the answer, but we will see. Yeah, in between it is mitochondria and microsomes. Mitochondria are smaller, but they are less denser than nuclei. Then microsomes are smaller and less denser than mitochondria. So A is the answer. Then next one. In a population, the probability of a specific individual getting infected with SARS-CoV-2 is low when a majority of individuals in the population become immune to the virus. Yes, that's correct. We have read it in papers during the COVID pandemic. So it is called as herd immunity. Answer is option D. Because of herd immunity, the majority of individuals in the population will become immune to the virus. Now what is innate immunity? It is a non-specific defense mechanism and it includes physical barriers, cell neutrophils, macrophages, complement system, etc. Then adaptive immunity, it is specific immunity and it is related with the T cells and the B cells. Then active immunity, that is a process by which individual develops immunity to a specific disease upon exposure to the pathogen. That is, upon exposure to pathogen, the individual's immune system, it will produce antibodies and will attack the pathogen as well as providing long-term protection. Then next one. Given below are 4 reactions of the glycolytic pathway catalyzed by the enzymes E1, E2, E3, E4 as indicated. Which of the enzyme is not part of the gluconeogenesis pathway? So glycolysis and gluconeogenesis, they can be reverted. If you reverse glycolysis, it is gluconeogenesis. But certain enzymes will change at specific reactions for specific reactions. See here, fructose 6-phosphate giving fructose 1,6 biphosphate, and even here in glycolysis it is catalyzed by phosphofructokinase. Whereas in gluconeogenesis, the reverse reaction, fructose 1,6 biphosphate to fructose 6-phosphate, it is catalyzed by fructose 1,6 biphosphatase. So they have asked which of these enzyme is not a part of the gluconeogenesis pathway. So E1 is not a part of the gluconeogenesis pathway. Now see E2. E2 fructose 1,6 biphosphate to dihydroxyacetone phosphate glyceraldehyde 3 phosphate. It is isomerase. So E2 is a reversible reaction. So the same enzyme catalyzes. Then phosphoenol pyruvate to pyruvate, that is E4. E4 in glycolysis, it is catalyzed by pyruvate kinase. Whereas this step, that is pyruvate to phosphoenol pyruvate, it is catalyzed by pyruvate carboxylase and phosphoenol pyruvate carboxykinase in gluconeogenesis pathway. So these two enzymes, that is E1 and E4, they do not participate in the gluconeogenesis pathway. But in glycolysis, it is the E1 enzyme and E4 take part. Gluconeogenesis, it is reverse of glycolysis. Then next one. Which of the following molecule is or are second messengers produced by the phosphoinositide signaling cascade? So the phosphoinositide signaling cascade, it involves phospholipase C activation, and this phospholipase C, it will cleave phosphatidyl inositol 4,5 biphosphate, that is PIP2, to two messengers, that is inositol 1,4,5 triphosphate and diacyl glycerol. So your answer here is, they have asked which of the following molecules is or are second messengers produced by phosphoinositide signaling cascade. So it is inositol 1,4,5 triphosphate and diacyl glycerol. Then next one. A protein has 7 cysteine residues. The maximum number of disulfide bonds of different combination that can possibly be formed by these 7 cysteine residues. So don't consider this disulfide bond I have drawn here when I am explaining first. Later I will tell you why I have drawn it. So first will be, this is the protein with 7 cysteine residues, 1, 2, 3, 4, 5, 6, 7. So this first one, it can form disulfide bond with 1, 2, 3, 4, 5, 6 residues, 6 cysteine residues, because disulfide bond require 2 cysteine residues to be present, and so one will form disulfide bond with 7. Suppose, okay, one will form disulfide bond with 7. So 6 for 1. Now coming to the second one. Now second, okay, after this disulfide bond formation, how many residues are remaining? 1, 2, 3, 4 and 5. Now take the second cysteine residue. It can form disulfide bond with 3, 4, 5, 6. That is 4 cysteine residues. So into 4. So that is the second forming disulfide bond with the 6th residue. Now how many are remaining? 3, 4 and 5. That is 3 cysteine residues are remaining. And they can form disulfide bond with 2 cysteine residues. That is between 3 and 5. So into 2. So the answer will be 48. Then next one. A lyophilized sample of 20 nanomoles of an oligonucleotide is dissolved in water and the volume of the solution is made up to 200 microliters. The concentration in micromolar of the oligonucleotide in this solution is. So first convert nanomoles to micromoles. 1 micromole is 1000 nanomoles, that is 0.02 micromoles. Volume they have given 200 microliter, that is 0.2 ml or 0.002 per 2 liters. Now calculate the concentration. It is number of moles by volume. So 0.02 by micromoles by 0.002 liters, that is 100 micromolar. Then next one. DNA is a 1 centimeter long chromatin, contains 5 into 10 raised to 9 base pairs. The fold compaction of this DNA within the chromatin is. So fold compaction, it is given by length of DNA molecule by length of chromatin. Now we have to calculate length of DNA molecule. We know one base pair is 0.34 nanometers. The rise per base, that is 0.34 nanometers. So 5 into 10 raised to 9 base pairs will be 5 into 10 raised to 9 by 0.3 into 0.34 nanometers, that will give you 170 centimeters. Now they have given length of chromatin as 1 centimeter. So 170 divided by 1 centimeter will give you 170. That will be the fold compaction. Then next one. Intracellular concentrations of ATP, ADP and inorganic phosphate in 4 cell types is given below. Which of these cell types has the most negative delta G for ATP hydrolysis? So I have given the explanation in the next slide. So this is the cell types ATP, ADP, inorganic phosphate. So we know ATP is giving to ADP plus Pi. Delta G is equal to delta G0 plus RT ln concentration of products by concentration of reactants. So here delta G plus RT is going to remain constant always. But here the product concentration of product and reactant changes. So substitute for concentration of product and reactants, then I will tell you what to do. So 5 into first one inorganic phosphate, 5 into 1.8 divided by 3. So ln 3 is equal to 1 for cell type L. For K similarly it is 3 into 1.3 divided by 3.9. So it is ln 1, it is ln is equal to 0. So here delta G is equal to delta G0. Now for cell type B, it is ln 2.7 into 0.7 divided by 2.7. So ln 0.7, it is a negative value, that is minus 3566. For cell type M, again ln is equal to 0. So delta G is equal to delta G0. So we know that for ATP hydrolysis, delta G0 is always negative. Okay, so this value is going to be negative. If, now listen carefully, if ln concentration of P by concentration of R is going to be positive value, if this is going to be positive value, then delta G, this one, delta G is going to be less negative than delta G0. Okay, if this is going to be positive value, so here it will be plus, here it will be minus, and delta G will be less negative than delta G0. Okay, now suppose if concentration of P by concentration of R is going to be negative value. Okay, if it is going to be negative value, then delta G is going to be more negative than delta G0. Okay, now see which cell type is having a negative value for concentration of P by concentration of R. Here it is for L it is 1, for K it is 0, for B it is negative. So B will have delta G more negative than delta G0. So your answer here will be cell type B. Then next one. Which one of the following amino acids has more than 2 acid base groups? So alanine, leucine, phenylalanine, they have only two acid base groups. That is, one is alpha amino group and the other is alpha carboxyl group. But if you see tyrosine, it has got a side chain which is phenolic. That is, it is having a hydroxyl group there. So other than alpha amino group, alpha carboxylic group, it is having a phenolic hydroxylic group. So there are more than two acid base groups for tyrosine. Now coming to the next question. Enzyme activity profiles as a function of time in the absence or presence of different types of feedback mechanism is shown in the figure below. Match the following feedback mechanisms with the corresponding profiles in the figure. So if you see P, no feedback mechanism, that is when you provide signal, activity is there. When signal is not there, activity not there. So it is no feedback mechanism. So P is 4. Then you see S1, positive feedback mechanism. So even when signal is not there, that is over here, this region, there is feedback mechanism. It is showing feedback mechanism. So it is positive feedback mechanism. So S is 1. So P4 and S1, that will go for option B. Wait. C also, it is P4 and S1. Okay, we will see Q. Q and R. Negative feedback mechanism with short delay. So Q must be this one. 3. There is a short delay. Then these two short delay and then negative feedback mechanism. So Q must be 3 and R must be a long delay. That is why it is like this. So it is 2. So answer is option B. Okay, option B is correct. Then next one. A linear DNA fragment of 5 kilobases when completely digested with EcoR1 produces 2.5, 1.5, 1 kb fragment. Complete digestion of the same 5 kb fragment with X bar 1 produces 3.5 and 1.5 kb fragments. Which one of the following sets of fragments will be obtained if 5 kb fragment is fully digested with EcoR1 and X bar 1 simultaneously? So if you see option A, it is 3 kb and 2 kb. Okay, they both add up together to give you 5 kb. But what about these fragments? 3 kb you are not going to get. It is 0.5, 3.5. Then 2 kb also you will not get. So A is not the correct answer. If you see B, it is 2 plus 1, it is 3. You won't get that. But C, you see. 3.5, 1.5 is given by X bar, 1.5 again. But here EcoR1 will not cut over here. So I am putting 1.5 here. Okay, then 2.5 and then 1 kb, it is given by EcoR1. So when you digest with X bar 1 and EcoR1, you are sure shot going to get a 1.5 kb fragment. But see here, it is 2.5. That can be split into 2 and 0.5. So X bar 1 will cut over here. So 2 plus 1.5 will give you 3.5, which is cut by X bar 1. Then you will have a 0.5 kb fragment and a 1 kb fragment. Okay. So if you see EcoR1 cutting for this double digestion product, it is 1.5 2.5 over here and 1 kb. That is what you got over here. Then if you see x bar 1 cutting, it will be 1.5 plus 2, it will be 3.5 and 1.5 by x bar 1. So C is going to be the answer. D, you are not going to get the 0.75 kb fragment anywhere. So D is also not the answer. So C is the answer. Then next one. Match the cell types in group 1 with associated processes listed in group 2. NK cells. NK cells, it is antibody dependent cell mediated cytotoxicity. So P3. P3 is there in C and D. B cells, they are for antibody production. So Q1, Q1. So C must be the answer out of C and D. A and B we are not taking into consideration because P is not given as 3. So Q1, C must be the answer. Then mast cell histamine production R4 and neutrophils, they are the first cell to be recruited at the site of infection. So C is the answer. Then next one. 4 statements about lipids are given below as option. Choose the statement which are correct. Cholesterol is amphipathic. Yes, it has both hydrophobic and hydrophilic regions. So A is correct. Self-assembly of phospholipids in water is due to hydrophobic effect. That is also correct. Here the non-polar tails, they will aggregate to minimize contact with water. So B is also correct. The temperature at which the gel phase changes to liquid crystalline phase increases with an increase in the degree of unsaturation of fatty acid tails. That is not correct. The temperature at which the gel phase changes to liquid crystalline increases with decrease in the degree of unsaturation of fatty acid tails. Because an unsaturated tail can disrupt the packing of the liquids. So C is not correct. The choline head group of lipid is positively charged. That is also correct because it has quaternary ammonium group in it. So D is correct. So A, B, D are correct. Then next one. Which of the following techniques can be used to separate proteins according to their molecular weight from a mixture of proteins? Ion exchange chromatography cannot be used because it separates protein based on their charge and not by molecular weight. Size exclusion chromatography separates proteins based on their size and the smaller proteins will elute last. So that is correct. Sodium dodecyl sulfate, polyacrylamide gel electrophoresis, SDS page also correct. Sucrose density gradient centrifugation, yes, that is also correct based on density and size. So denser proteins will sediment faster. So B, C, D is the right answer. Then next one. B cells produce two forms of immunoglobulin, membrane bound form known as B cell receptor and the soluble form known as antibody. Which of the following statements is or are correct about BCR and antibody produced by the same B cells? BCR and antibody have identical antigen binding sites. That's correct because they are derived from the same variable region genes. So they have the same identical antigen binding site. BCR and antibody recognize different epitopes. They recognize the same epitope. So B is not correct. BCR and antibody are encoded by the same gene. That is correct. Then BCR and antibody are formed by differential splicing. That is also correct. So A, C, D is the answer. Then next one. A 100 ml solution of pH 10 was mixed with 100 ml solution of pH 4. The pH of the resultant 200 ml solution. Basic chemistry question. So pH is equal to 10. pH plus pOH is equal to 14. So pOH is going to be 4. Concentration of OH is 10 raised to minus 4 moles per liter. So in 1000 ml you have 10 raised to minus 4 moles of OH. So in 100 ml, that is what they have taken. So it is 10 raised to minus 4 into 10 raised to 2 divided by 1000. So it is 10 raised to minus 5 moles. Similarly, calculate hydrogen ion concentration for pH 4. So that will give you 10 raised to minus 5 moles. So total amount of hydrogen and OH minus ions in 200 ml. It will be 0.5 moles divided by 200, that is 5 into 10 raised to minus 5 molar. And concentration of OH minus also will be 5 into 10 raised to minus 5 molar. Since concentration of H plus and OH minus are equal, the solution is neutral. So pH is equal to minus log concentration of H plus, that is minus log concentration of 5 into 10 raised to minus 5 molar, that is 4.30. So the answer is 4.30. Then next one. An organism uses only glycerophosphate shunt pathway to transport cytosolic NADH to mitochondria. For every 2 electron transported, complex 1, complex 2, complex 3, complex 4 of the electron transport chain in this organism transport 2.5, 1.5, 2 protons respectively. So this is complex 1, it transport 2.5, complex 3 it transport 1.5, complex 4 transport 2. Then the H plus to ATP ratio, that is for ATP production, the protons will move into the mitochondria. So H plus to ATP ratio of the F0, F1 ATPase of this organism is 4. So 4 H plus ions are transported inside. So terminal electron acceptor is oxygen. The number of ATP molecules synthesized by oxidizing NADH from glycolysis is. So first you have to see that glycerophosphate shunt pathway, it generates FADH2 in the mitochondria, which donates electrons to the complex 2 by passing complex 1. Okay, remember that. Then, so the total proton pumped per electron pair, that is for 1 FADH2 it is 1.5 plus 2. We will take into consideration only complex 3 and 4. So complex 3 it is 1.5 and for complex 2 it is 2. So it is 3.5 H plus. Now during this glycerophosphate shuttle, 2 FADH2 are generated. So 3.5 into 2, that is 7 H plus. So H plus ATP ratio they have given 4. That means 4 protons are required to synthesize 1 ATP molecules. So 7H plus will synthesize how much ATP? That is 7 by 4, that is 1.75. Then next one. If the extracellular concentration of sodium ions is 10 times more than its intracellular concentration, then the sodium equilibrium potential at 20 degrees Celsius in millivolts is. Assume that the membrane is permeable only to Na plus ions. Okay, so you have to use this equation. E is equal to RT by NF into Ln concentration of Na plus out by concentration of Na plus in. N is 1, the other is given because sodium ions, it is plus 1 plus. So it is 1, N value is 1. T convert to Kelvin, that is 293 Kelvin. Substitute the values, you will get it as 0.05801 volt. Convert to millivolt. They have asked in millivolts. So it is 58.01 millivolt. So thank you for watching my channel. If you want GATE Excel 2024 question papers answered, I have already done that. You can refer my channel Omni Bio Entrance for the same. Thank you.